So what I want to do is to show you the major toxigenic orders and examples within those orders, and then have a few samples for you to look at. And then, as I said, I've got some keys. I don't have 40 keys, but I've got 15 or 20 keys, so you can work. Or if you don't want to look at run the keys, you don't have to. You can just look at water samples. But let me just summarize then these toxigenic orders, because I really haven't done that. We talked about the taxonomy, but we really haven't talked about the practical side of things. Well, there are harmless algae, and of course, being able to at least visually recognize when you should be more concerned about whether this is a cyanobacteria, these are some of the things to avoid. Because on the left, this is one of the more harmless, filamentous, stringy-looking, probably Cladophora in this case, would be my guess. Might be Spirogyra. So that's a harmless algae. If you can pick up and they don't fall apart, they stick in your hands like a mat, they probably are not cyanobacteria. Okay? And looking at the mat on the right as well, there's a certain look to that. You will see really greeny green looking blue-greens, but blue-green blooms tend to have an off-green color to them. They aren't bright green like green algae mats. So, you know, again, side by side, it's easy to see, and then I can tell you that, but it's generally true that the cyanobacteria bloom will be not a bright green, it's more of a dull green in color. Likewise, I've had samples sent and said, this is a cyanobacteria, it's dangerous, and this is a duckweed. Maybe you've had the same experience, and that's fine. You know, don't laugh, just smile and say, no, I think you're fine, you're okay. Duckweeds are, in fact, vascular plants. And in fact, at certain times, you get nice little flowers that they produce as well. But now here's a bloom, and there are just different look to it. Sort of an off green, not a bright green, more of a dull green, more streaky, more like the term has been used like an oil slick or something, or a paint slick. It really does describe cyanobacteria more than it did another kind of algae bloom. Okay, the toxigenic. And I use the term toxigenic to imply that they are capable of producing toxin. It doesn't mean that they are always producing the toxin. But toxigenic cyanobacteria are found in 4 of the 7 orders within this new classification, the Cyanoprokaryota. And I'm not going to bore you with the new taxonomy. I'm just going to tell you that there are now 7 orders of Cyanoprokaryotas, and 4 of those 7 orders contain members that are toxigenic. Okay, and the main classification, those 3 big books that I mentioned on the taxonomy, Yuri Kumarek has actually condensed down in 2014 into a nice little thing that's 40 pages. So if you have to have something, get this paper right here. Summarizes it all and has nice tables and everything. So it's really the better way to kind of have at your fingertips the current taxonomy without having 3,000-page volumes. So that's taxonomic classification of Cyanoprokaryotes. And it's all of them. Yes, the filamentous ones, the unicellular ones, all of them are summarized in that 40-page paper. Now, the main taxonomic changes from the old system to the new system that you're going to have to accommodate at some point because it involves a different term. Planktic, meaning planktonic or floating in the water column. Members of the genus Oscillatoria are now Planktothrix. That's been around a few years, and I think most people have adopted that. It used to be that these straight-chained filamentous ones without specialized cells of heterocysts and aconites were very often an Oscillatoria. But what they've taken and reclassified and put all of the benthic members as Oscillatoria, and the planktic members are now Planktothrix. implying they're in the plankton, floating in the plankton. Secondly, the planktic members of the genus Anabina are now almost completely a new genus called Dolichospermum. And I don't like it, but I'll accept it. So I did my thesis with Anabina, so it's always going to be Anabina to me. And in fact, I was given an Anabina right here. See, this is Anabina. And there's the heterocyst. Yeah, there's a company that makes several of those little stuffed animals in the form of microbes, not just Anabina. Thirdly, the planktic non-colony forming Aphanizomenon is now called Cuspidothrix. And I say that because there are a few members of Cuspidothrix that also produce a toxin. And in one particular, Toxigenic is a saxitoxin producer, which we identified a number of years ago. But the toxic species Gracile is still in the genus Aphanizomenon, so this becomes a little confusing. So you're going to have old Aphanizomenon with two different genera now, some producing toxins. But again, the description of that is all in that little 40-page paper, Humorex. So let's look quickly. The first order, Chroococcales, family Microcystaceae. Obviously, this is going to be your microcystin producers, Microcystis. And so the main one is species aeruginosa. There are about 6 or 8 species of Microcystis. I've seen some of these, but for example, 1 and 2 is aeruginosa. The middle one is actually a species called botrys, and the bottom is firma. And it's just based upon that colony shape. Now, my advice to you is, don't worry about the species. All Microcystis potentially produce toxin. Now, aeruginosa is probably going to be producing a toxin all of the time, or almost all the time. Some of these others won't, so you'll have to run a test. But they all can produce microcystin.
So aeruginosa, its genes will always be turned on?
Pretty much, yeah, pretty much. I don't think I've ever seen one that isn't. If it isn't, you know, the gene is not showing expression, it's probably one of these other species. A couple of others then, top Microcystis flos-aquae, Microcystis ichthyoblava, and Microcystis novosecchi. I'm sure you're going to memorize these, but you're going to see different colony shapes of Microcystis. And I just want you to understand that they are still Microcystis. Pseudofilamentosa and Smithii, another one. This one's pretty obvious. These are really long, hundreds of microns long for a single colony, but quite narrow and shaped pretty much like that. Well, that's why they're called pseudofilamentosa. It's like Harry Potter's, you know, magic expressions, they sound like. Viridis, pretty much, Viridis produces toxin almost all the time as well. Wiesenbergi, the same, pretty much always a toxin producer.
Is there a table about the toxin producing aspects of each of these?
Not really, no, because so many of them haven't been tested. This is just kind of my sense of doing a lot. And in fact, this paper is produced by a Chinese lab studying these species. And if you really want them, I can get you the copy of the paper, but this is a colleague of mine, actually one of my postdocs who now works in the Hydrobiology Institute in Wuhan. Secondly, the order Nostocales, family Aphanizomenonaceae. These are the main producers of cyanotoxins. All known cyanotoxins are found in this family. Dolichospermum, or Anabina, couple of different ones here. There's semi-coiled ones and the straight chain one. These are both. This is Dolichospermum, formerly Anabina. Species flos-aquae is this one, and this one is a different species, Polyplanktonica. Now you can see the heterocysts here, but you can also see aconites in here. These sort of light, elongated ones, bigger than the heterocysts, are aconites. Those are the spores, those resting spores. I don't think you'll see those in the samples today, but. And here's, this is pretty much what you're going to see in the two samples I brought. This is from a lake that I've been helping a group with. It's called Cullowhee Lake. It's over on the coast near Astoria. And two species seem to dominate. Both of these are Dolichospermum or Anabina, Crassum and Planktonicum. And Planktonicum, I'm pretty sure you have in Clear Lake, right, as well? Maybe. We'll see. So the more coiled one is the Crassum, and the straight chain one is Planktonicum. And again, here, wherever you see a light dot, that's going to be one of those heterocysts, but I don't see any aconites there. So that's the one that I'd like you, the sample I'd like you to sit down with and walk through the key and prove to yourself that it's Anabina. So I'm telling you the answer, so see if you can walk through the key.
But it's not, it's Dolichospermum.
Okay, Dolichospermum, yes. I told you I refused to. Aphanizomenon itself, in this case the species flos-aquae, you can see the giant, see how big that aconite is? Those are huge. That's one of the key characters of this species. And I put this up because this is the one harvested for a food supplement at Klamath Lake. It, of course, washes down the river and grows in the reservoirs. But in the reservoirs, it tends to be displaced by Microcystis, so the reservoirs on the river, that's why they're trying to take the dams out on the river for that, and for fish. But it forms these flakes, and the filaments are straight. But again, nitrogen fixing. And this particular species of Aphanizomenon is non-toxic, which is good for the industry but there are species of Aphanizomenon that are toxic, but not flos-aquae. If you haven't ever been to Klamath Lake and seen the harvesting operation, here's just a couple of pictures. They drag these screens and screen the algae up and dewater it and refrigerate it, dry it, process it, and then make you healthy. There are about 5 harvesters of Aphanizomenon on Klamath Lake, but they barely make a dent in the biomass. I mean, 0.000%. The hundreds of thousands of pounds that they harvest each year is a very small fraction of what the standing crop would be on that lake.
So is it okay to just point out, we actually had a post Upper Klamath Lake last year for Microcystin being above Oregon's threshold. So
almost every, yeah, that's a good point. Almost every year, and that's one of the things I've been doing over the years, is making sure that they have monitoring programs in place, quality control programs in place, that they don't harvest and they don't have algae. And they've done very well. Whenever the Oregon State Health Department has tested the algae, it may be positive, but it's always below 1, which is 1 microgram. Well, it is for this company, and I know that they have exceeded it, but not in the recent future, and not in this company. But as I said, there are 5 harvesters on the lake.
But you said they're testing for Microcystin because
of Microcystis. Microcystis.
Not because of the AFA.
No, the Microcystin produced by Microcystis is what leads to the posting and the non-harvesting. So they actually have microscopes on board. They have ELISA 2 kits on board that they monitor, and they just don't harvest if it's exceeded. Plus they're now, this actually, digressing, but this little harvester is designed to harvest only Aphanizomenon. The screen mesh size is set up so that the colonies, the round colonies of Microcystis, actually slide right on through. and it removes up to 99%, and it always manages to allow them to stay below the 1 microgram per gram regulation.
So what do you think that does to the prevalence of Microcystis in the lake? Is it increasing the impact?
Not really, because keep in mind they're only harvesting a very small fraction of the entire biomass. So even though it's not like Lake Erie where all those zebra mussels are selectively putting Microcystis back in the lake, that does make an effect. This doesn't. They're just using such a small amount of the algae. So that's one way that they're able to harvest longer, even though this doesn't give them very much. Literally hundreds of pounds a day instead of thousands of pounds a day, which this harvester can do.
So the Aphanizomenon is always the dominant?
Yes. This was why they can have an industry there.
About what percentage of Microcystis would be involved in a bloom?
Anytime the Microcystis population begins to exceed 1% of the cell counts, you'll probably have toxin levels exceeding the 1 part per billion. So again, that's something that's worked out over time.
I thought the blooms were kind of offset, the AFA and then...
Well, they are, but they like to harvest year-round if they can. So right now, the blooms are just starting, and it'll be dominant Aphanizomenon. By the end of July, Microcystis, or even in July, Microcystis might start to show up. And they'll either have to move their harvesting to different parts of the lake, or they'll have to shut it down. Typically in this lake, Aphanizomenon blooms very heavily in July and in August, and often late August, early September, there's a crash of Aphanizomenon, and then the harvesting's over. But then it comes back in October, and they'll actually go back and harvest in October and November. There's a cycle to the Aphanizomenon. But again, it's only because this lake is rather unique, producing Aphanizomenon, at least since the 1930s. And there are very, very few lakes in the world that you can rely on having the same bloom. I mean, not even Clear Lake can you go and say, okay, this year is going to be the same as last year. There are very, very few lakes in the world that so it's a bit of a unique ecology. The north side of this lake has a lot of underground artesian spring, because Crater Lake is literally just 40 miles away. And that 3,000 feet of water, 2,000 feet of water, actually percolates and creates a lot of underground springs that wash into the rivers that feed the upper parts of this lake. And those rivers are very good fly fishing rivers as well. So.
Well, doesn't the AFA have that aconite, so it's called, so it's constantly reseeding itself?
Oh yeah, right. Yeah, so on this lake, the phosphorus is coming from the volcanic soils, and nitrogen is limited, hence you select for the nitrogen fixers. So the concern I have is that if you don't try managing that upper system, the watershed system, because there's like 10 or 20,000 cattle are put on that upper system every year out of feedlots, and then they come back to California for the winter. They're just summer feeding. That you're going to have phosphorus inputs that are going to at some point start pushing that Microcystis to dominance as opposed to Aphanizomenon to dominance.
There's a lot of efforts that they've been looking into. You know, some of the new agreements are with riparian corridors, and I'm part of a group that's looking at different treatment techniques, and one of them is that has a lot of promise at this point is diffuse source treatment wetlands, kind of like scattering wetlands across the upper watershed.
A couple of the bigger farms, one in particular, about 3,000, was purchased by the Nature Conservancy, and they're starting to breach dikes and flood land and sort of to absorb those nutrients, but there's still a lot of cows out there. Okay, sorry, we're on to the genus Genus Cylindrospermopsis, producing cylindrospermum. I'm sure you have this in California. The thing about this is it's a straight chain filament, and it has these terminal aconites that are very pointed. That's a real giveaway to that particular genus. The tapered terminal aconite. And then the lighter dots in between would be your heterocysts. You can see from this angle, maybe there. That's cylindrospermopsin. Now, again, remember there are other things producing cylindrospermopsin, but that's the type genus for it. The order Nostocales, family Stigonemataceae. Example, Stigonema is a non-toxin producer. So I just wanted to throw that in. Here's an example of those filaments that are branched. See how the sheath grows and then the cell bends and kind of looks like branching. So this is false branching, but the genus Stigonema, and it's not a toxin producer, but it's in that. And there are other members of that group that do produce toxins. The order Oscillatoriales, the last one, family Microcystaceae and family Oscillatoriaceae, does have toxin producers. This group doesn't have any of these secondary, these accessory cells, no heterocysts, no aphanites. So here's an example, the genus Planktothrix on this side, straight chain, no specialized cells, so that's how you would tell it apart from Dulicospermum. The cells are also slightly smaller, and that would be defined in your key as well as you work through a dichotomous key. The one on the right is a benthic oscillatoria that produces microcystin or could produce saxitoxin as well. This actually comes from Rafe Cadella's lab picture. And the genus Lyngbia I've thrown in. This is also in this group. This one can produce those contact toxins, Lyngbia toxin. But I like this one because it shows very well the sheath that surrounds this and how you can actually break the filament. And of course, if this was to grow, you could have false branching. Now, Lyngbia doesn't do that, but some do. And that's why it's Lyngbia. It doesn't. These are fairly big. These cells are quite large. Notice that the scale, 30 microns, so they're about 30, 40 microns across. Maybe 5, 10 times the size of the microcystis cell. And Microcoliaceae in that same grouping, the genus Arthrospira. I threw this in because Arthrospira is the new name for spirulina, and spirulina, of course, is the other major cyanobacteria used as a food supplement. And so the picture on the right is these pills, capsules with the spirulina powders, and I like spirulina better than Arthrospira, even though Arthrospira implies spiral, spirulina sounds more spiral. And this, it very much is a filament. Again, no heterocysts, no aconites, but a very tight spiral organism. Okay, in summary, the toxigenic members are found within 4 orders, 11 families, 22 genera, and about 40 species out of about 1,500. So there's your task. So let's go across the way and have a look.